All right, so mitosis is something we expect you to know, we expect you to be able to draw. We're telling you that this cell starts in interphase. That's where we're telling you to start the cell cycle. And when we draw a cell in interphase, we have a cell, a diploid cell, and it's going to have some genetic information. Key things, what phase or what form is the genetic information in? Chromosomes or chromatin?
Chromatin.
Tin, bowl of spaghetti, right? Does it have a nucleus or no nucleus? It has a nucleus. So really important things that we draw. I'll draw a nucleus on this thing. Okay? And we draw DNA in the form of chromatin, which we can easily just make some squiggly lines and say, hey, look, it's in the chromatin form. Very, 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 very big picture. We start with one diploid cell, and we end up with how many? 2, and are they haploid or diploid cells? Okay. One parent cell, we end up with, and what's true about those 2 daughter cells?
Genetically identical.
They're genetically identical. Okay, so if we started with a diploid cell, in order for a diploid cell to be genetically identical, then we have to end up with 2 diploid cells, right? So very big picture, we start with one diploid cell, we end up with 2 diploid cells. Di meaning 2, and really the idea of a diploid is you end up with, you have homologous pair of chromosomes, you get one from mama, and you get one from dad. I don't know why I said mama and dad, I never say mama and dad, from mom and dad. Okay, your biological mom and your biological dad. During sexual reproduction, if you're sexually reproducing, I should say. So we have chromatin. Okay, is interphase part of mitosis?
No.
No, it is not part of mitosis. There are 3 things that there are 3 phases of interphase, and in the order that they occur, they start with G1 followed by S followed by, and during G1 the cell, and during S the DNA. I led you probably too much with that DNA part, but the DNA replicates, and then G2 we grow a little bit more and prepare for replication. Okay, G1, S, G2. Sometimes the cell will go into G0, sometimes it comes out, sometimes it doesn't. Okay, now we start the mitosis process. And our puppy mitosis needs a PMAT, so it doesn't go to the bathroom all over the place. So P, M-A-T-C. So P for prophase, M for metaphase, A for T for telophase. Now you have some artistic freedom, I think we could say it that way, for those last 2 boxes. There's 2 different kind of approaches you could put for those last 2 boxes. For the second to last box, you could call it telophase and telophase only. And if that's true, then what would the last box need to be labeled? Cytokinesis. Or in that second to last box, you could go telophase / cytokinesis. And if in the second to last box you put telophase / cytokinesis, that's okay. What would the last box need to be then? What comes after cytokinesis? In a cycle, we go back to interphase. So there's some choices you have there, okay? We'll get to that. So prophase, PMAT, prophase. DNA is in what form? The X form or the chromosome form or the chromatid form, but we often draw chromosomes. Is that nucleus intact or is it breaking down? Breaking down, it's dissolving. So we draw a dissolving nucleus and we draw chromosomes. And if we say, hey, I'm going to draw 4 chromosomes, this is where we get homologous pairs, and we can, the color coding purposes, we get a big and a little, and a big and a little. 2 different colors. Okay? It's nice to have a big and a little compared to all the same size, and it's really hard to tell which ones are the pairs of each other. So we have red and green on Mr. DeWar's picture. Okay, but we have chromosomes. The two bigs are called homologous pairs. It's not super critical for mitosis. It's going to come in handy when we start talking about that stuff tomorrow with meiosis. So we get the starting of spindle fibers. Not critical, not huge. It's something that we can kind of, in the back of our mind, keep in mind. Meta and, oh, it'd be good for you to make sure you have a corrected copy. That's why the we draw column is there, okay? Meta, middle, meta, middle. What goes to the middle? The chromosomes go to the middle. Okay, your choice, equator or prime meridian, and how you're going to make this division happen. Your choice, whether it's horizontal or vertical. Okay? Looks like Mr. DeBoer is going equatorial.
And it's going
Okay,
to...
so we have 4 chromosomes. We have 4 chromosomes. Okay, and he put them along the equators because they go to the middle. Meta, middle, meta, middle. Why did they go to the middle? What pulled them to the middle? The spindle fibers. Okay, those spindle fibers pull them towards the middle. It's in chromosome form. Notice how we have X's drawn, right? Meta, middle. That's the important thing for metaphase, is those chromosomes are in the middle. Meta, middle, middle, meta. Questions before we flip it over?
can you wait a minute, real quick?
We shouldn't need to wait too long here. Meta, middle.
I'm almost done.
After meta, we go to anaphase. Ana, apart. Ana, apart. Apart. Ana, good. Okay, what pulls apart in anaphase? The chromosomes. And when we take X chromosome, not an X chromosome, that sounds like we're talking about X and Y gender chromosome. When we talk about a chromosome in the X shape, what pulls apart? What do we call 1/2 of the X? What? I thought I heard it. Nope. The chromatid pulls apart. Okay? And the other half is called a chromatid. And collectively, the 2 chromatids that are held together by a centromere in the middle are referred to as sister chromatids. And what's true about those sister chromatids? Genetically identical, because when was DNA replicated? In what part of interphase?
S?
During the S phase of interphase. Good. So in Ana, things get pulled apart. So those chromosomes that we had aligned in the middle get pulled apart. So those X's get pulled apart. So half of an X goes, the way Mr. DeBoer is drawing it, north, and the other half goes south. We have 4 of them. Those 4 get divided in half. Those centromeres break down. Those sister chromatids are pulled apart. Notice how in Mr. DeBoer's picture, he showed that the cell starts to elongate, right? Yesterday that was in that color sort. We didn't draw it originally, but the idea is when those pull, they're going to kind of get more oblong shape. Okay? Now, again, you have some choice. Do you want to go telophase / cytokinesis, or do you want to go telophase cytokinesis?
Phase and then cytokinesis.
Okay, so if we do it that way, that's fine. The thing that's a little tricky about this is during telophase, I really like the slash personally.
I can change it.
Let's slash it. Your choice though. Okay, you do have the choice. The thing about cytokinesis is what happens in cytokinesis. Nope, the nucleus reforming is telophase. What's that? Yeah, we get the pinching, right? It's the cytoplasm and the cell membrane pinching together. I ran out of one, Mr. DeBoer, so it's over here. Okay, so if you wanted to draw telophase separate, it's okay, but then you should have 0 pinching. Okay, if you draw telophase separately, you should really only have the nucleus reforming and your DNA going back into chromatid form. By drawing it kind of in the pinched format, by slashing it, okay, we can then go through and we can kind of get both things going. Now to talk briefly in that color sort activity, they talked about a cell plate. The cell plate is really the start of a new cell wall. That's what it is. A cell wall on a plant is rigid, made of cellulose. It doesn't pinch very well, right? It's very pretty. Rigid structures don't pinch very well. So while the cell membrane will pinch, the cell wall doesn't. So a cell plate is really the formation of a new cell wall, but it's kind of from an inside-out growth. So we have DNA that is split. We have nuclei. Oh, Mr. Kunin, I am so sorry. I forgot. I forgot to tell him. We have the nucleus starting to reform, and we get the pinching of this cytoplasm. I was supposed to tell you this before. He came looking for you, and I'm like, can you have him come down? I'm like, absolutely.
That's all right.
He popped. Okay, so we get the cell membrane pinching, we get the nucleus reforming. Now that DNA is going to start to uncoil. That's also the process that starts happening in telophase. Final product, we need to end up with how many cells? 2 cells. So they can be bigger or smaller than what we started with. Smaller. And are they going to have a nucleus or no nucleus? Are they going to, yes, nucleus. Are they going to have chromosomes or chromatin? Tin or tid? Which one did you say? D or N? N. Chromatin, which is uncoiled DNA. Bowl of spaghetti again.
I like spaghetti.
Okay? So in the final process, so the way we just drew this is we did cytokinesis with telophase. So then our final box would be interphase. It's acceptable, absolutely acceptable, to go through and say those are 2 separate phases, and you have the pinching going on here. I kind of like it this way because then we can say interphase, we end up with 2 diploid cells, right? Now there's a few bullet points on the bottom, and the 2 bullet points on the bottom are to go through and kind of major things. So the parent cell, is the thing we start with, is genetically blank to the daughter cells, and we already mentioned this one, but just so we get it in paper, they're genetically identical to the daughter cells. Because the sister chromatids pulled apart, and sister chromatids are genetically identical to one another. Mitosis is a process that is done by what kind of cells go through mitosis. Give me an daughter is what we call them when they're done. Give me an example of a type of cell that goes through mitosis in you.
Body cells.
Yes, that is the general classification that I was looking for. Is body cells. So what's an example of a body cell? Skin cells, right? What else? Bone cells. Good. Bone cells, someone.
What about bone marrow?
Which is a type of...
So because technically you get more.
Hair cells, yes. Muscle cells, yes.
Blood cells?
Blood cells, yes. Okay. How about sperm cells? How about egg cells? No. Those are called sex cells. That's tomorrow. We're talking about those tomorrow. Okay. Body cells, which is all things in your body, with the exception of what we would call sex cells, which is egg or sperm cells in humans. We call those sex cells. 1 diploid. Diploid means a complete set of chromosomes, 1 from mom, 1 from dad. How many X's did we start with? Did we draw in prophase? How many X's did Mr. DeBoer draw in prophase? 4. How many chromatids are in the top cell of cytokinesis / telophase? 4. 2 of them are green, 2 of them are red. 2 came from mom, 2 came from dad. That makes them a diploid cell. They have 2 bigs, 2 littles. That's a diploid cell.
2 bigs, 2 littles. Can you flip back to the front for metaphase real quick?
Alright, with the little bit of time we have remaining, I'd like you to return your colored pencils. And we're gonna get started on this quiz, and I sure hope we finish this quiz before we leave today. All right, so there's a quiz on mitosis that, again, you are going to take. We have about 20 minutes, okay? 20 minutes. There's 17 questions, kind of, sort of, because there are multiple parts of some of these. But hopefully we get these done and finished before we walk out of here today. Okay? So work on this quiz. Hang on to them for the time being. We'll give you instructions on what to do with them when we're all done.